So hi, everyone. It's very nice to meet you. My name is Dr. Natalie Lambert. I'm an associate research professor at Indiana University School of Medicine. And for the pandemic, it seems like a very long time now, but since the earlier days of the pandemic, I've been collaborating with Survivor Corps, which is a patient group for primarily long haulers. She can't hear me. Let's see. I'll turn up a little bit.
I can hear you. Yeah.
Okay. And I've been working with them to try to capture patients' experiences with the disease. And I'm just going to talk a little bit to sort of set the scene for long COVID in case some of you aren't as familiar with all of the symptoms of it. But we have a panel here with two other fantastic speakers. So it's nothing too formal, but just to have a conversation and even answer people's questions. Back in the beginning of the pandemic, before we really knew very much about COVID, it was very much characterized as a type of flu. So early on the CDC's website, when they talked about coronavirus, they were saying, if you have it, you should look out. If you think you might have it, the symptoms to look out for things like fever, cough, and shortness of breath. After that, it was expanded to other symptoms like muscle or body aches, loss of taste or smell, vomiting, diarrhea, all of that. Well, the way that I conduct my research is primarily by going online and collaborating with patient groups, and especially Facebook groups. And Diana Berent had founded a Facebook group for people who had COVID and an amazing job of bringing people to that page and over time mobilizing them to support each other. But what we found early on in that group was that many people who got COVID weren't recovering, and they were having a very wide range of very unusual symptoms that one would not expect from a flu. So things like fatigue, headaches, what patients describe inability to exercise, memory problems, heart palpitations, what they call like different types of painful nerve sensations, and the list goes on and on. And, so we've been working very hard to get the research done to try to capture these experiences, but connecting with other researchers and physicians, we found that a lot of these symptoms mimic the symptoms that people with other chronic conditions also suffer from. So these types of connections between people who study diseases that can arise from other types of viruses has been really essential for understanding long COVID. And sort of where the state of a lot of my research is right now is trying to understand health impacts. We know that there's a lot of symptoms from long COVID that can make it very difficult for people to work. We know that a lot of this wide array of strange symptoms have the tendency to come and go, that they can be exacerbated by exercise, that rest is a big component of it. We know that there's problems like vascular damage, neurological impacts, inflammation is a big part of it, many different suspects of the key mechanisms of how all this is happening. And we're doing research to learn more every day. Another study that I completed recently was trying to understand problems that patients experience when they're seeking care for long COVID. And what we're finding is that not having an official diagnosis for COVID, so let's say you got sick very early on in the pandemic before there were tests, or let's say you got sick later, tests weren't as available, or maybe your physician didn't encourage you to go get a COVID test, not having that official COVID positive test in your medical record can make it very hard to access care. And there still is a long way to go to making sure that physicians have enough knowledge about long COVID to be able to recognize it and not attribute it to things more like mental health impacts. Especially we're looking at with COVID with children. Children can become long haulers and get long COVID in medical records. They pop up in pretty high percentages. So it's another area we need to do a lot more research in, is how COVID is impacting kids, and make sure that when pediatric patients are presenting with these unusual symptoms, to not assume that it's not due to COVID, but to look at the physiological signs of that. So that's sort of a very broad introduction. I'd love to pass it on to some of my other panelists so that they can give their perspective and talk a little bit about the work that they're doing for long COVID. Can I pass it to you, Stephen? Is that okay?
I think you're muted, Stephen.
I was trying to be a good boy, muting myself. Sorry. Can I move myself up, or do I wait for someone to move me up to that speaking position?
Let me do help here. There. Go ahead. You can move up now.
Okay, thank you. Hi, I'm Dr. Stephen Phillips. I'm a physician who's been researching chronic illness and chronic illness patients since 1990s. Really, I've been in practice since 96. I've treated over 20,000 patients with complex chronic illness. My practice focus has historically been vector-borne illness, and other insidious infectious diseases, some parasitic infections as well. And I'm the co-author of a book, Chronic, that delves into the challenging aspects of these chronic infections. And we have a chapter on COVID because the overlap between long COVID and the patients that I've been seeing for the last 25 years has been eerie. There are some stark differences, particularly between the high rate of vascular complications with long COVID and less so with the patients that I see. But these other symptoms of headache and cognitive problems and fatigue, especially post-exertional malaise and heart palpitations and neuropathic pain and weird sensations that you would go to the doctor and they would chalk it up to anxiety, things like internal vibration sensations. I've been seeing these types of symptom complaints in my patients forever. So when COVID came around, I started noticing some of my patients who had gotten COVID, even mild cases, the character of their illness started to change. And they went to a few doctors and some of them got diagnosed with long COVID. And when they came back to me, upon treating these patients with antimicrobials for, let's say, underlying infections like Lyme and Bartonella, I would notice that many of their symptoms attributed to long COVID would flare up, would get worse during treatment. It's a phenomenon that we see called a Herxham reaction, which happens with particularly some bacterial infections. And I'm wondering if some immunologic aspects of COVID in some cases are changing the landscape of how we deal with other chronic infections that people don't readily, think about. So everyone's heard of Lyme disease, and most people fight about Lyme disease, but very few doctors even talk about infections like Bartonellosis. And these are infections that share a lot of the same overlapping, difficulties that we see with COVID. There are problems with testing, there's problems with definition of cure, there's problems with chronicity. And I'm seeing the character of these problems, like I said, change. And we're starting to see long COVID patients, de novo, patients who don't carry a diagnosis of these other infections. And many of these patients are responding to antimicrobial therapy, again, with Herxham reactions, worsening of underlying symptoms, then improvements. And you say, why is this happening? How is this happening? Before COVID came around, we would see many, many patients who their story was that they underwent a bad viral illness. They had a bad flu, or they got dengue, or a bad case of mono, and then they just never recovered. So the question is, does the virus persist, or is it upsetting the immunologic apple cart and allowing other infections that were asymptomatic previously to become symptomatic? And the take-home point is that the infections that we deal with, things like Lyme and Bartonella and many others, the asymptomatic to symptomatic ratio is quite high. It's definitely more than 2 to 1. So we think that there's a lot of asymptomatic infection in the population, and the question is what happens to these people when they get COVID. So that's all I would say as part of my introduction here. So I guess Amy, if you'd like to talk next.
Sure. Do you want to move me up?
I don't know how. Okay.
Hi, my name is Amy Perez. I'm a microbiologist with a nonprofit research organization named Polybio Research Foundation. And I guess when long COVID began to be documented in patients were reporting chronic symptoms after acute COVID disease, we were not surprised because our organization had already connected a number of research teams to study patients whose conditions are also initiated or exacerbated by infection. One of them is called myalgic encephalomyelitis chronic fatigue syndrome, or MECFS. That's a condition in which the majority of cases begin with another viral infection, a herpes virus infection, or an enterovirus infection. And enteroviruses are from a family of polioviruses. They're also, like SARS-CoV-2, single-stranded RNA viruses. And so there have been outbreaks of MECFS in which clusters of patients infected with enterovirus, a subset have gone on to develop chronic symptoms. And in fact, there have actually been researchers, for example, that have found chronic enterovirus in either the brains or stomach biopsy specimens of patients with MECFS. And then beyond that, most major pathogens, almost all of them, from Zika to Ebola, from dengue, and as Steve mentioned, Borrelia, Bartonella, are associated with a certain subset of patients who get that pathogen, go on to develop chronic symptoms that don't go away. So this is a phenomenon that we've known about a long time before. So unfortunately, we actually suspected that this would happen with COVID. And when we were building on that, along with my collaborator, Mike Van Elzeker, he's at Harvard MGH, we wrote a paper on long COVID, and we in the paper delineate several multiple scenarios that may lead to development of long COVID. And to do that, we already pulled from the what we call infection-associated disease literature, the post-Ebola syndrome literature, the ME/CFS literature, and other parts of what we already were learning about the SARS-CoV-2 virus. And yes, briefly, one of the first most obvious things that we think could be happening is that the virus doesn't fully clear in all patients. It may very well be persisting, at least in some. There are some studies that have looked at tissue samples from patients months or weeks after disease, or at least months, and have still found the genetic material of the virus or the proteins that it creates in those tissue samples. The real catch there is that I mentioned tissue samples. If you're going to study that topic of SARS-CoV-2 persistence, you need to collect tissue, or you need to do autopsy studies, because the virus is seldom in the blood. Most viruses are seldom in blood or body fluids over a longer period of time. In acute disease, they might be there, but over time, they, in simple terms, will hide in tissue or a nerve. So you have to get those samples if you want to figure out if they're still there over a longer period of time. But then a second scenario we talk about in our paper alludes to what Steve mentioned, in which SARS-CoV-2 will take down parts of the immune response in order to better survive even when it drives acute disease. So it creates at least 10 proteins that downregulate interferon signaling. Now, interferons are proteins that they're antiviral cytokines that keep other viruses in check. For example, the herpes viruses. So if you have your interferon signaling knocked down, that's a great opportunity for Epstein-Barr virus, HHV6, cytomegalovirus to reactivate in a patient and themselves drive chronic symptoms. And 90% or more of the population harbors these viruses. They're usually in a latent state, but COVID may give them an opportunity to activate, infect new tissue or nerve themselves and cause disease. And the same goes for the bacterial pathogens that Steve mentioned. For example, some people get a tick bite and they honestly do not notice it. They get Borrelia, they get Bartonella, they have these infections. Again, if the immune system can somewhat contain them in a lot of people so that the symptoms that they have, even though they harbor these organisms, don't manifest. But acute COVID may serve as, again, a source of stress that dysregulates the immune response and allows those bacterial organisms to also, again, drive disease in and of themselves. And this may be not, different patients may have different things going on. We don't think there's likely to be one long COVID. We suspect that there's mixing and matching of these different phenomenon. And our paper even goes into other things, coagulatory issues, microbiome and virome dysbiosis in different body sites. And we're interested in how all of that can impact signaling of how the vagus nerve is able to signal to the dorsal brainstem, which controls a lot of autonomic function, flu-like symptoms, and other symptoms that manifest in patients with long COVID. So that's my intro.
Thank you. Do we have another speaker? Maybe we should open it up to questions. Remember, you have to unmute yourself to ask a question.
So I have a question, if I might. What do you think would be the key directions forward in terms of doing research, doing care? What are the, you've described most of the current conditions. What do we want to have as next steps?
Should I take that question? Okay, let me start on that, and then I'll try to also, I hope Natalie and Steve can jump in as well. That's a great question. So the way our organization is looking at it is we first want to explore that first possibility that SARS-CoV-2 does not fully clear and persist in some patients. So what we're trying to do is we're trying to set up a tissue biobank of both post-mortem tissue collected at autopsy, and also we're trying to collect surgical or biopsy samples that contain tissue or nerve from patients with long COVID. Then we're going to analyze those samples for a range of infectious, genetic, and immune abnormalities. In order to do that, we do use a lot of the latest, and I think it's important, sort of computer-based techniques, whole genome sequencing, metatranscriptomics, shotgun sequencing, spatial transcriptomics. We can really, really find organism in a sample using those computer-based tools in a better way if you do it right, and depends on the methods, than we have been, have been able to for many years. In other words, it's exciting that the ability to look for organism in tissue also sort of is concurrently happening at a time when what is called the tissue microbiome is becoming a very prolific area of research. So for example, tumors, people thought they were sterile, or maybe that one virus, and some viruses like the herpes viruses are oncogenic. But now there are teams that are turning whole genome sequencing, which is a computer-based method of looking for organism genomes in a sample, onto tumors. They're filled with bacteria. They're filled with viruses. There's multiple organisms in there. They're all interacting. There's bacteriophage in them, which are viruses that infect the bacteria and modulate their activity. So there's a whole lot of stuff going on in tissue, and we definitely need to explore that in patients because SARS-CoV-2 infects cells via the ACE2 receptor, which is dense in tissue. So if we really want to find what it's doing and what happens in that tissue, whether it's other organisms, whether it's the human gene expression in that tissue, we need to do a lot of analysis of tissue. And the next thing I think that we can correlate that with, and what we're trying to do, is really intelligently done neuroimaging on the same patients. Because we need to understand what's going wrong with their brains and bodies in order to best understand what the pathogen or may be doing, and honestly, what tissues to prioritize for study. So we're trying to do some neuroimaging studies, and we're already doing them on patients with ME/CFS, where we can do autonomic challenges in the scanner. For example, we do a breath hold test where just holding a patient holding their breath leads to a challenge in which there's a carbon dioxide oxygen shift, and you can watch perfusion of blood to the brain. In ME/CFS patients, the ratio of blood perfusion to the brain can't rebalance correctly. So we can immediately say, blood perfusion in the central nervous system doesn't seem to be happening. Then I can say, it seems like the blood vessels or vasculature may not be functioning. That gives me clues. I say, we need to look at tissue samples containing blood vessels, right? We can also do another neuroimaging kind of challenge where we use a vagus nerve stimulator in a scanner. And we can use that stimulator to stimulate the vagus nerve in patients, which is a very important nerve that connects most of the trunk organs to the brain. And we can say, is that signaling going correctly in these patients? If it isn't, then we can say, let's look at vagus stimulation. And then from that, we can build in and say, what therapeutic approaches can we use to try to address this? If we're finding the virus in tissues, can we use some of the antivirals that may come out on the market in long COVID? I don't know. Can we improve the immune response to better, part of it may be that patients with long COVID are not clearing the pathogen because the immune system is not that great. Maybe antibody signaling could be better. If it involves vagus nerve signaling problems or vasculature issues, how can we intervene? How can we do that? And last, I'll just do a plug for clotting and coagulatory issues in long COVID. There's one team we're working with. They're finding microclots in simple terms and hyperactivated platelets in the blood of patients with long COVID. They really are. There's almost all the patients have these clotting issues. And that's not disconnected to the possible persistence of SARS-CoV-2 or reactivation of other pathogens, because platelets, these red blood cells, they contain receptors that sense viral and bacterial proteins. So as soon as they sense something like the SARS-CoV-2 spike protein or a herpes virus protein or a Borrelia protein, they activate. They become activated. They signal fibrinogen, which is a clotting molecule, starts to bind with the platelets, and you'll see a bunch of coagulation begin. Once the blood becomes coagulated, it has trouble circulating through the body. It may not reach the peripheral nerves, which are the small nerves that can lead to symptoms that can cause neuropathy. Those nerves may die off. We already see in patients with ME/CFS and related conditions that those patients have a high incidence of small fiber neuropathy. That may be a problem then, where the blood cannot reach the small fiber nerves. That leads to the fact that those nerves may be more easily infected. So we can begin to combine some of those trends. And that's kind of what we're looking at.
Great. Thank you.
Can you guys hear me?
Sorry?
Yes.
Can you hear me? Okay. I'm echoing. It's Dana. Sorry, my connection is so poor, but I wanted to ask Steve a little bit about early treatment and what you found to be helpful and what you think is the best option or the best options on the table as we know it now. I'm really interested in early treatment, and I think you should talk about maybe how important that is in general in infectious diseases and COVID.
Okay. I mean, so in infectious diseases, and just as a standard, you don't wait until you cut your finger till it becomes a big streaky cellulitis going up your wrist before you treat. The outcomes are worse. And it confused me at the beginning when there were some, albeit, small, albeit sparse, albeit not the best designed studies of very, very safe options. Ivermectin has become this like hot topic, controversial drug now, whereas before I had over a decade experience with ivermectin because it has immunomodulatory effects and immunopotentiating effects, and I've used it in my chronic illness patients with great benefit. Around 2018, we started using it in MS patients because there's some data showing that it could induce remyelination. There's also data that it induces a permissive state for peripheral neuropathy, damaged peripheral nerves to regrow. So ivermectin has all these effects that I've been, using for my patients for years. And when it came out that it may have activity against COVID, I had such a great comfort level with it and so much experience with its safety that I didn't feel at all uncomfortable about trying it in these patients. And during the time that alpha strain occurred, I'd say, my practice is outside of New York City. We had about close to just under 100 acute patients we treated. And all my patients have chronic illness. That's what the practice consists of. And on top of that, many patients had traditional risk factors, obesity, hypertension, diabetes, heart disease, etc. And not to sound cocky, because I'm not cocky about Delta, but alpha wasn't a problem. I mean, I treat patients, I give them doxycycline, ivermectin, and a bunch of supplements like quercetin and vitamin D and zinc, and patients turned around quite well, and nobody progressed out of close to 100 patients. Like I said, even patients with high risk factors. Delta's been proving more of a challenge, and I don't think ivermectin is a panacea or anything like this, but I still think it's a useful tool. We're also offering patients fluvoxamine. There's been recent data about reduction of mortality by roughly 90%. More data I heard is coming down the pike on that one. But again, we have, these patients that have multi-systemic presentations, we have lots of experience with fluvoxamine as well, because a lot of patients get neuropsychiatric aspects to their illness. Inflammation of the brain, it always struck me as odd that people would either view things as psychiatric or neurologic and not both, because when the brain gets inflamed, people can get numbness and weakness and tingling. And they can also get agitation and irritability, depression, anxiety, and nuances of things like OCD. And we have seen in my patients, it's a good 40% of my patient population gets new onset of neuropsychiatric illness. And when they get treated and their physical symptoms, their psychiatric symptoms go away as well. And if I didn't see it every day for the past 25 years, to talk about this to a general internist, they say, that sounds unusual, except the fact that it's been published repeatedly in the literature, and it's not really common knowledge. So fluvoxamine is a good option. We're also very pro monoclonal antibody in the first few days. Earlier the better, and we've seen very good outcomes in patients who are at high risk. We give them monoclonal antibodies and turn them around. And in terms of later treatments, I think that all bets are off because we don't have really reliable, good later treatments for COVID. So we focus on the early stuff. I'm very excited about the anti-androgen therapy that they're looking at. They're now expanding the trials to look at females as well. There's already been good trials about anti-androgens in men with good reductions of clinical illness and survival, and those are our main options that we offer patients.
Dana, we can't hear you, I don't think. Can you check your connection, Dana? No.
Dana, are you muted?
She's not muted, but we can't hear her.
Okay. So, if nobody else has any questions.
Steve, I feel like she was probably asking you how you also treat the Lyme Bartonella quickly. I think that was part of her original question. Like if
quickly?
It turns up. Yeah. I guess a few quick points on that.
5 minutes. I mean, so the issue with Lyme is that there's been there have been volumes of data showing that B. burgdorferi persists despite antibiotics. They're supposed to be curative in animals and in people. And the problem with fastidious organisms is that they're fastidious. And, you know, the definition of fastidious in microbiology is that they're not readily cultivable. They can't be grown easily in routine culture media. So if you inject B. burgdorferi into a dog and then you let the dog get sick, you know the dog has Lyme, you just gave him Lyme, and then you take blood from the dog, nothing really grows. And it's been very, very difficult to prove anything in this field because of the difficulties cultivating, you know, these types of pathogenic spirochetes. So similar things, similar problems are present with Bartonella and Brucella and Coxiella and, you know, the bacteria that causes tularemia. This is not like a Borrelia specific issue. There's a number of fastidious organisms out there and doctors don't understand them. But the most well-known of them is Lyme, and that gets all the heat and all the controversy. But the fact is that the studies vary. Most studies average around 25% of patients when they receive a kind of a standard course of 3 to 4 weeks of antibiotics with early Lyme will go on to develop long-term symptoms. Some studies have shown, you know, less, but those are actually few, even though it's frequently said that roughly 10% go on to develop chronic symptoms. Johns Hopkins did a study, it was 39%. Danbury Hospital, which is the largest, you know, private registry of Lyme disease in the U.S., they did a study, and it was 61% of patients had the identical symptoms a year later who received a full month of antibiotics for erythema migrans, which is a Lyme rash. So, and there are, you know, there's really good animal data where they treat all sorts of animals, whether it's rodents, dogs, horses, and even monkeys, our closest, you know, human model, with the antibiotics that are purported to cure Lyme, and the animals are they're not being cured. On top of that, we can't effectively kill this bacteria in the test tube with the antibiotics that are purported to be curative. And lastly, there is human data. And like Amy said before, it's not so much blood cultures. Very hard to find in fluids, easier to find in tissue. They have found it. They have found this organism alive despite literally years of antimicrobial therapy in patients. And yet, despite all this evidence, despite everything that's out there, including the NIH doing a study of ticks where they put uninfected laboratory-reared ticks on patients with what they were terming post-treatment Lyme disease syndrome, some of these ticks got infected with the blood from patients with post-treatment Lyme disease syndrome. So I do think that persistence is a mechanism by which pathogens, you know, they evolve this over time. And like she said, there's evidence that SARS-CoV-2 does persist, at least in some studies. I also agree they're not looking in the places they need to look. You can't tell what's happening in the rest of the body from one drop of blood. And Dana, you had messages that you're back on.
I think you're muted now, Dana. Still can't hear you. You can write something in the chat if you want.
Let's see. Where's Natalie?
She was here.
Okay. Just want to make sure she gets enough time to talk to you.
Yeah, I can get out of this hot seat, or if I get myself out.
Oh, no. That's what
I'll
you're
do
saying.
it.
Okay.
Can you hear me now? Can you hear me now? Yes.
Now I hear you.
Okay,
So
go ahead.
My
Dana,
question
talk.
is for Natalie, and I want to know if there is a subset of patients that is more prone to long COVID. If that's the case, what is that group of patients? What do they look like? And what are the most prominent symptoms that you're seeing at this time? And has it changed throughout the course of the pandemic with the different strains of COVID? Is Natalie still here?
I don't see Natalie in here. She was here for most of this. Let's see. Let's see if I can find her.
Okay.
No, Natalie is gone apparently.
And
Sorry,
did we lose
we lost
Steve?
Natalie.
No, we lost Natalie.
Steve, are you here? Yeah, Stephen is now in the row that is not is muted. So let me just grab him. Stephen.
I could also try to answer your question, Dana.
Yeah, I'm really interested in that. I'm so curious about that, and I think a lot of people want to know what is really going on.
I mean, Amy, you want to tackle this one? I'll get out of here.
Sure, whatever. Should I move myself? Or wait.
I couldn't seem to move myself effectively.
We can hear you.
Okay. All right, I just moved myself. Okay. Dana, so you were talking about why do some people get long COVID and others not?
Yeah, I'm curious as to whether there is a certain health status or certain characteristics or list of characteristics for people who are more prone. Is it still women in their 30s and 40s that change and evolve? Or is there no pattern at all?
Right. So, I think that when you're looking at any chronic disease, you first understand that if you look at most of the diseases now and the way they're progressing, there's an interaction. There's usually human genome variants in the human genome that can predispose to problems in the patient. And then there's the manner in which different parts of the environment act or compound those problems with the human genome in order to really exacerbate the disease process. So for example, in Alzheimer's, APOE is a gene that can predispose towards Alzheimer's risk, but herpes simplex 1 virus can augment the gene expression of that gene, right? So a popular and interesting topic of research now is actually herpes virus activity in the Alzheimer's brain, because it may be a mix of genetics and infection that it can exacerbate the disease process. I suspect that there's at least some of that trend in long COVID, where I think it's important to identify human genome variants that may already set people up. So for example, we were talking about that patients with long COVID may have coagulatory issues. Well, there are already genes in which people are predisposed to increased clotting. So if you already have that gene, you may be more prone to coagulation driven by the SARS-CoV-2 spike protein, right? Now, of course, though, we're all also, and I think this is a really important thing to think about, we're all seeded with with interacting communities of bacteria, viruses, fungi, and like I mentioned, bacteriophage, viruses that infect bacteria. There are trillions, if not quadrillions, of these organisms in most of our tissue and blood. And when I say quadrillions, I'm actually referring to those bacteriophage. The current estimate is that a quadrillion or more phage, this is coming from Paul Bolsky's team at Stanford, traffic our tissues at any given time. And we now understand that these organism communities are not just in the gut or the mouth where they were originally identified. There's a lung microbiome environment. There's organisms in the bladder, in the pancreas, often in blood. An organism can leak from the body sites like the mouth and gut that harbor the most organism into the blood under conditions of inflammation or imbalance. So for example, if you get COVID, your immune system is thrown off, it's dysregulated, it's trying to fight that infection. That provides an opportunity for organism, let's say in other body site, for example, just the gut. The barrier or permeability of the gut can become leaky in simple terms, allowing more organisms to get into the blood. That then can cause its own, it can activate cytokines, the bacterial products will drive inflammation. So where, some patients may already have some of that going on before they get COVID. And I think that sometimes if you talk to long COVID patients, and I've done this for years with patients with ME/CFS, if you really listen to their stories, they'll tell you, I did get much sicker after COVID with chronic symptoms, but before that, I'd had some GI issues, or I had problems with this before, or sometimes I felt weird, and so there may be in some cases where, let's say, a person already had what we call imbalance or dysbiosis of the gut microbiome. And so there was more leakage of organism into blood. So when they got COVID, there was already more bacteria in blood, which themselves can stimulate, for example, clotting processes. And then you add in the SARS-CoV-2 virus, it does something similar. And those are the ones where it really takes off in, right? And for example, that can be with the collective activity of organisms like I mattered, or as Steve has alluded to, just with single pathogens. If you happen to already have Bartonella infects the vasculature. It infects blood vessels. So it may be lingering in some blood vessel. Now, SARS-CoV-2 also really infects the vasculature and can drive problems with the vasculature. So if you already had Bartonella and then you got SARS-CoV-2, it's like a double hit on the vasculature. So you may be one of the people who could go in a direction in which vasculature issues become problematic in your case. So those are some possible forms of predisposition. And again, what I would say is collective microbial load, single pathogen load ahead of time, and also human genome risk are at least some factors.
I'm wondering if people who are treating long COVID, are they doing extensive co-infection testing, say, the way that physicians who take care of Lyme and tick-borne patients do?
Steve, or do you know? I would say that before Steve gets on, currently the studies that are being done on long COVID, I'm not sure I'm seeing that being pursued too often. One of the reasons is it's hard to do. For example, let's say you just want to know if there's increased herpes virus activity in patients with long COVID. The herpes viruses are, again, rarely in blood. Again, you might need to look for tissue samples. You can sometimes look at the immune response, for example, an antibody response to something like herpes virus in blood, but it can be difficult to do, and the testing isn't great. So one of the things that we need to do is improve the diagnostics to be able to identify these other organisms, which have actually it's been deprioritized in research up until this point, I'll be honest. I do think that many people have been pushing for better ways to identify herpes virus activity in the human body, all kinds of other pathogen activity before COVID started. And I actually think that it wasn't until COVID when people actually started to realize that the testing for COVID, for SARS-CoV-2 itself, was really not that great, was not that reliable, often was, hard, really not perfectly indicative of the disease process, that people began to say, is that the same case for herpes virus testing, or is that the same case for Lyme testing? And it is. So what it really means is that collectively, we need to improve our diagnostics and the manner in which we evaluate patients for not just SARS-CoV-2, but a broad range of pathogens and organisms, and that perhaps COVID would give an impetus for more research to be done on that topic. Steve? Yeah, can you hear me? Okay. Steve, do you have any other thoughts?
Am I still muted?
No, there you are. Right.
Okay, so I couldn't talk when I was down there. Can I talk up here? It was hard for me to hear Charlotte's question. Hi, Charlotte. But it sounded like you're asking if there's evidence of co-infection in long COVID patients. I'm not aware, like Amy said, I'm not aware of, this is important data that, research needs to be done. In my practice, we have cases, like I said, where patients were diagnosed with long COVID and they clearly responded in the way of getting Herxheimer's, which shouldn't happen with a viral infection solely. Not saying that SARS-CoV-2 is not present in their body, not saying it's not causing coagulation issues, not saying it's not doing damage, not being neutral on the whole thing, just agnostic. But from the aspect of other chronic infections, these people have had Herxheimer's and improvement. We had a guy come in who could barely walk, and with relatively simple innocuous therapies has markedly improved very, very quickly. And I'm like, why isn't anybody, looking at this part of the research, but this part of the spectrum? I think it's difficult because it's a hard thing to sell. Like Amy said, that we don't have a sterile internal microenvironment, that there are microbiomes everywhere, including the brain and the blood. We've done whole genome sequencing of patients' blood just to see what we can find. We've found a whole range of organisms that we think most of them are red herrings, but we find them at various, labs, both private and academic, and we don't really know what to make of them except to say that we are, our bloodstream is seeded regularly by organisms. And are they coming from when we brush our teeth? Are they coming from microenvironments in the tissues and sometimes going out? But I do think it's an area that needs to be studied. I think that one of the weaknesses that we have, we study things in a way that's not how real life works. We don't study networked environments. We study pretty much, single variable infections or maybe 2 infections at a time, but it's very difficult to study. I mean, they're starting to get more, computerized stuff where they study the interactome, but it's a network solution that we're looking for, and we're looking at it in this really overly linear way. It's not how real life kind of works. So that's my answer.
Thanks. And I think we have a question in the chat from Christine. She wants to know, are any of the long COVID centers looking at and testing for persistence and treating?
I'm not sure if the long COVID centers are looking at referring persistence of SARS-CoV-2, because I haven't heard of, very few of my patients have gone there, but the ones that have weren't tested in any way. They didn't get tested for like GI biopsies or olfactory bulb biopsies or anything like that.
Thanks. Does anybody else have any questions? I can unmute or go ahead.
I was going to say there's 1 or 2 long COVID treatment centers that I think are opening to the idea of trying to collect patient tissue samples for analysis of SARS-CoV-2 over time. But it's definitely, I'm not sure if it's prioritized in the NIH's main long COVID program. With that being said, then that's called the Recovery Initiative. They haven't really announced what they're doing yet very much. So it's sort of open question. And I do think that if you look at historically speaking, a lot of times it's difficult to get, IRB approval to collect those samples. It's difficult to stabilize those samples. So there's some teams that won't do it because it's actually just harder to do. But part of what our organization is doing is trying to help some of the long COVID centers understand that we really do think it's important. So I think it's an evolving topic at the moment.
Thank you. Anybody else have anything? Let's see. Christine asked again. What are the long COVID treatment centers offering to long COVID patients?
Well,
okay.
No, go ahead.
No. I mean, depends on the long COVID center. I did, there's a center at Sinai in New York, and they are, doing their best to offer rehabilitation type therapies in which patients with dysautonomia can do some treatment protocols that can sort of help with dysautonomia symptoms, also breathing exercise type protocols. But I would say that what they're trying to do is they're most focused on sort of what I'm going to call injury related sequelae. And of course that can be happening in these patients. I think that's one of the things that with long COVID that, is that some patients, long COVID is a broad term right now. Like, what does it mean to have long COVID? There are clearly some patients who had a very bad case of COVID. They were in the ICU. They had the virus, just beat up their lungs, their heart. They just really ravaged their bodies. So those patients probably do have ongoing injury, fibrosis, lung, there are certainly a category of patients like that. Those are the ones that the long COVID clinics are helping somewhat, treatments for fibrosis, improved lung issues, definitely a focus on the lungs, which makes sense because it's COVID. I mean, that's where they're going first. So, but I don't think that first of all, necessarily, it takes a while. We've been in what I call the infection associated disease field for a while. It takes a while to really realize the longer term symptoms that some of these patients develop that are not injury related. So what a lot of long COVID patients go on to develop are similar to the ME/CFS patients, are similar to the post-treatment or chronic Lyme patients, and that's a mix of autonomic dysfunction, relapsing, remitting flu-like symptoms, malaise, and then also a cardinal symptom called post-exertional malaise, in which if they even exert themselves in a minor fashion, physically or even mentally, just trying to process something in a difficult cognitive fashion, it actually flares all their symptoms and they crash, they become much worse. Now that is a symptom that develops like down the line more, or whatever, in the patients that are probably less of the injury cases and more of what is this phenomenon of sort of infection associated disease we've seen with other viruses over time as well. And that has been an issue, understandably, in a sense, with some of the long COVID clinics now, Because a classic, especially in USA medicine, when you find a patient, they seem sick, they seem like they need help, you go, get back out there. Maybe we should have you exercise a little bit. Maybe you should get out and keep trying to push yourself, especially if the patient, and this is the thing, you can get the long COVID symptoms are so diverse that some patients do have depression, they do have anxiety along with all these other symptoms. And as Steve mentioned, we literally understand that pathogens can drive neuropsychiatric symptoms. I mean, I could give you dozens of examples of that. For example, toxoplasma is a parasite that actually more than 11% of the USA population harbors in the central nervous system. And if it becomes active, it can drive schizophrenia type symptoms. I mean, we know this. So it doesn't necessarily mean that those symptoms have to be dissociated from an infectious process in these patients, but some do have anxiety, depression symptoms. Really, when it comes to those symptoms, those patients that the medical community thinks, get out there, exercise, get out of this, you're depressed. So there's been a learning curve with some of these long COVID clinics where they really had to see that those patients, a lot of those patients have instead this post-exertional malaise where they cannot do exercise, difficult exercise programs. So a lot of their iterating right now has been now on focusing on small pacing protocols where patients are actually try to stop activity before they crash and trying to manage crashes and trying to manage pacing, which is definitely helpful for symptom management, but it doesn't necessarily get to the root cause of the problem, but at least it helps patients cope.
So can I say something? Yeah. So I've been unimpressed with the work thus far from the small amount of patients I've seen from COVID centers. I always say you have to get to the root cause for things. It's the whole premise of our book. And my patients who are quite ill don't very much benefit. Just before COVID, just looking at the chronic illness community before this, patients with severe cases of ME/CFS have historically never benefited from intensive exercise and retraining programs. It really hasn't helped them. And it's the patients, like Amy said, that had this significant insult to their body that are slowly healing over time and don't have a waxing and waning course. I don't think those patients necessarily have to be evaluated for other occult infections that could have been reactivated. But the patients who develop long COVID after a 2-month kind of, a quiescent period after COVID resolves, acute COVID resolves, or the patients that have a waxing and relapsing or remitting kind of a course, those patients I think very much should be evaluated for what else can be going on in addition to the presence of SARS-CoV-2 still in the body. I don't think they're mutually exclusive. There's evidence of some of this persistence of this subunit spike protein. Where does that subunit come from? I don't think that proteins just persist in the body without source to have them persist. It's an analogous situation where they looked at Lyme, chronic Lyme arthritis, where they found peptidoglycan. Peptidoglycan is a component in, bacterial cell membranes. Where's that coming from? They're saying, it's just persistent peptidoglycan driving the chronic Lyme arthritis. Again, the body's very good at getting rid of foreign material. It doesn't just hold on to them forever. It digests it and gets it gone. So if these foreign antigens, these foreign, materials that are stimulating the immune system are still there, then there's going to be a source for their continual production. And so, I'm disappointed that the centers, as of yet, maybe they'll change soon, but as of yet, I haven't seen much of looking for cause. And whether it's SARS-CoV-2 by itself as the cause, or whether there's immunologic perturbations that are predisposed to the reactivation of other microbes. I did a talk at MIT a couple months ago, and I started out by saying that there's 1 trillion microbes they're estimating on the planet, and we've yet to discover 99.99% of them. And we think we have it all figured out. We don't even know. Science has not even identified more than 99% of the microbes we harbor in our own GI microbiome. So I think we should feel humbled before this and just be neutral and let the science lead us and go in the directions that make most sense. And I don't know I don't think a one-time insult and, oh, you just have to recover makes sense for those people who have this kind of asymptomatic period after acute COVID, then get sick with long COVID, then have this up and down. To me, there's some active infection going on. We've got to figure out what that is.
Okay, I think we're just about out of time, but there was one more question, if I can sneak it in, from from Dana, about the Pasteur Institute study. Do you want to talk about that?
Dana, you talking about the olfactory bulb stuff?
Yes, that's what she said.
Yeah, so no, it's just a study where they found, evidence that is highly suggestive of active viral replication in the olfactory bulbs of patients who had persistent, anosmia, lack of smell. I think it was like 6 months after acute COVID. So we know that it can persist, but we've already known that coronaviruses can persist. There's evidence of coronaviruses in the brains of MS patients, not implicating in this case that coronavirus is causing MS, but just saying that we know that it can persist in our bodies. There's evidence. I don't know if you guys ever heard of the case of some researchers that were overwintering in Antarctica, and after being down there for many weeks, one by one, half of the group down there got the common cold. And it just goes to show how little we know about the common cold. Where they get it from? They were down in Antarctica by themselves with nobody around, and one by one they start getting the cold. So obviously, did one of them harbor the cold in a way that we don't think even a common cold virus should be harbored? We think of it as a short incubation period. You get the cold, it goes away, and you spread it on. But even that, we don't understand properly.
Thank you. Does anybody else have any questions? Yep, excellent panel. Just reading some of the comments. Okay. And does anybody else have anything? I think we can go a little bit longer if the speakers are game, because we don't have anything else in this space butting up against us. So it's up to you.
I'm good if anyone has questions. Otherwise, I just want to thank everybody for your attention and inviting us to speak.
Excellent. Anybody else have any questions or comments? You're welcome to continue to explore this OEA space and network in the lounge areas. The share hall is where you can find, if you want to see what other talks are going to be starting soon, that's where you can find that. And thank you very much for your attendance.
So
great talk.
I have a question. With all of the need to get early treatment, why do you think they are still saying things like rest at home? It's going to be a mild illness, maybe get monoclonal antibodies, but really just rest.
Well, I think that the doctors, they risk stratify by, by age, by body mass index, by other underlying conditions. So they may give different advice to different people. A 70-year-old obese person with hypertension and diabetes, they may not say rest at home. But even young people can develop long COVID even with mild illness. This is the other big question. Does early treatment not only improve mortality outcomes, does it improve chronic illness outcomes? In my experience, it does. My patients, by and large, that have been treated early have not developed chronic symptoms. And that's what I've seen. I have the one patient whose character of underlying symptoms has changed. And I don't know what we would call her. We call her, she responds to antibiotics, which COVID, wouldn't. She responds with Herxheimer's. I think her Bartonellosis has changed. But none of my other patients have developed an increase in long-term symptoms when they've been treated. It's the patients who are away from the practice that may still harbor some infection in their body. They've been treated, they feel 95% better, they're back at work and everything seems good. They get COVID, don't get treated, and they come running back to the practice and they have chronic symptoms at that point. So I do think early treatment most likely cuts down on long-term COVID complications. And your question, though, was why do doctors say rest at home? I mean, I'm not one of those doctors. I think that you got to take the bull by the horns, so to speak. And I wouldn't say just rest at home. We know this virus can do a lot of bad things, and mortality is just one of the bad things that it can do. Chronic illness is the other bad thing.
Okay, and I think Eddie had a question. Eddie, can you unmute yourself? Or do you want me to ask it? Eddie can't hear you. Let's see. She wants to know if meat is safe to eat concerning COVID.
Is meat from what aspect is that? I'm sorry, I'm confused by the question.
That's okay. She says sausage, egg that is crude protein, maybe? Eddie, can you unmute yourself so that we can hear? I'm going to move her up again. Let's see. Now try, Eddie.
Yeah, I am unmuted, but it's
okay.
not.
Yep, we can hear you now.
Ah, okay, great. So what I mean is, if this virus would persist in the bodies, and we cannot be sure that farm animals are not, actually infected with it, would it be possible for this virus to transfer via meat? For example, sausage that is crude, right? I don't mean by fomites that somebody touched it with, that's something else, obviously. But I mean like just in meat,
right?
If we don't cook it.
If you don't cook it. So you're talking about cured products that aren't cooked?
Exactly.
I'm actually not sure. You know, the topic of foodborne transmission has come up quite a few times, but I don't think there's any consensus or big documentation of it. It's hard because, you know, so many cases are presented with primarily GI symptomatology. So you think, could this have happened? But I don't know how it's a very slippery topic, you know, and how do you prove these things? I guess they'd have to do studies to find out if the curing process, I personally doubt that it would happen, but I don't know the data, so I don't want
to. I mean, like ham, for example, right? It is processed in a way, so actually, there should not be. Like, it's not a optimal environment for a virus because it's drenched with. I mean, but still, we cannot know really if it hides in tissue, how it could be.
Right. The only way would be to test it. My sense is that it probably would not survive that curing process, or the salting process, and everything that they do. But I don't want to speak up with authority if I don't know. Amy, do you have an answer to that? Because I don't really know the answer to the question. Amy, can you hear me?
I think Dana is suggesting maybe Charlotte. Do you want to say something?
Charlotte?
You have to unmute.
So here, let me move Charlotte. I don't know if I have to move Charlotte up or.
I'll unmute. I'm not aware of any studies.
Okay.
Was that?
Yep, we can hear you.
I just want to say, I think that was an interesting question. You know, I don't know, but I can say that, for example, when we're thinking about doing autopsy work on patients who might still have SARS-CoV-2, we have to be careful to do the work in a BSL-3 lab because the virus may still be virus. So I don't know. I do think it's interesting, and it's not mentioned often, that other animals are getting sick with SARS-CoV-2. For example, I think there's a deer outbreak at the moment. I hope I'm being accurate with that.
It's true.
Yeah. So there's sure a lot of things to study, but that is actually, when you first asked the question, I thought, what? And then when you explained it, I thought that's a good question. Something we should think about.
Thanks.
Thank you. Do we have time for another question from the chat? Stefan is asking, suppose somebody just recovered from a COVID-19 infection. Is there a possibility that the virus would still be present and start manifesting later on after some weeks or months? Is that possible?
Yeah, it does seem possible. I could review a couple more of the just studies out there. where people have actually looked for SARS-CoV-2, the genetic material of the virus or protein in patient tissue months after initial infection. There's the olfactory bulb study that was mentioned before, olfactory bulb being a definite interesting area to be infected because olfactory bulb actually, there's neurons in the olfactory bulb that can connect to neurons in the brain. So other viruses can get into the brain often by infecting neurons in the olfactory bulb and then slowly moving into the back of the brain. So that's an interesting site for persistence. That was a small study. There were not that many subjects in the study where they found the virus there long term. I think we should be looking at more of those samples. But also there was a team that got, I think it was in Singapore, via surgery, they got colon tissue, ileum tissue, hemorrhoid tissue, tissue from, colonoscopy. I'm trying to think what other tissue types. Appendix, gallbladder, lymph node. That's interesting. And it was about 9 to 180 days after initial infection, and they found the viral genetic material and protein in the majority of those samples. Another team infected monkeys with SARS-CoV-2, and then when they looked at their tissues at autopsy, in 50% of the monkeys, the virus was still in the heart, the respiratory tract, lymph nodes, salivary gland tissue. And what was interesting about that study is you can look at what's called subgenomic messenger RNA, which actually is a type of RNA that indicates replication of virus. And they did find that subgenomic messenger RNA in the lungs and lymph nodes of the monkeys, which indicated that in that case, it seemed like there was ongoing virus replication. So those are two examples of studies that just have found SARS-CoV-2 long-term. Now, does that mean it's happening in everyone? I don't know. But, and then, I would add that there have been a couple teams, including one at Massachusetts General Hospital, that tracked what just is referred to as chronic SARS-CoV-2 infection in immunocompromised patients. So, it actually is completely accepted that patients who are, unfortunately taking immunosuppressive drugs for other, autoimmune type conditions, that they have trouble clearing the virus. So they tracked a patient, I think, for well over 100 days, and they actually tracked how the virus, it's an interesting paper, the virus mutated in that same patient to evolve new mutations. So it actually its genome changed over time in the same patient while they continue to harbor the SARS-CoV-2 virus. There's been 3 studies, I think, that have documented that in patients who are immunocompromised. And, you may say, well, those patients were very immunocompromised, and they were, but a large part of the population is immunocompromised. One of the things that's difficult is most of the medications that we give to patients with a range of conditions are immunosuppressive. So patients are being treated for autoimmune type conditions with a range, from Humira to this to other immunosuppressive drugs on a fairly regular basis. They are the most prescribed medicines for illness right now. So there may be, at least, a low level issue in that, in which if the immune system cannot be robust enough, that is probably another instance in which the virus may not fully clear. So it's not, I've always tried to encourage root cause findings in chronic disease for that reason, because if we don't look for pathogens themselves or for changes in organism microbiome in patients with chronic disease, and we only do analyses of the immune system, which happens often. What happens often in research is that there's a diagnosis, long COVID. The majority of the teams that jump in, what they do is they get a blood sample. And they study the immune cells. They study the immune parameters, the T cells, the B cells. And when they see that those are activating differently, or there's different cytokines or different immune cells, they go, the immune response is off. And they blame the immune system. And they don't say, why is it off? And the why may be the pathogens, the microbiome, this. So they tend to then make it a disease that's more an immune disease and give people immunosuppressive drugs. That is not great during a pandemic like this, when a new virus is infecting a lot of people. So you have to consider that a decent number of people are immunocompromised, and that is not a great situation to fully probably clear the SARS-CoV-2 virus.
Thank you.
Have there been any studies looking at, for instance, say semen, the potential sanctuary site for as an analogy depicted it in Ebola.
Good question, Charlotte. And Charlotte's referring to research in post-Ebola syndrome is a really interesting, if you're interested in what we're going to call post-infectious disease, look at research on post-Ebola syndrome, because patients get Ebola, a certain number of them develop really debilitating chronic symptoms, and for a while it was regarded as a mystery, a little like long COVID now. What's going on? What's the mystery? And then researchers finally began to look in what are called, they call anatomical sanctuaries, similar to a reservoir. And in breast milk samples, in semen samples, they started to find the genetic material, the RNA of Ebola, months or years after initial infection. Or the eye, for example. But semen and breast milk were two sites that were really common in terms of harboring the genetic material. And even then, a lot of people in the research community said, oh, well, that's just RNA. It's just the genetic material. It's not the virus itself, which is honestly an argument I don't understand, but it was kind of like, oh, it's the RNA. I don't know. So that kind of percolated for a while, that these patients just have RNA in those body sites. But then a team published a really good study in the journal Nature where they showed that like levels of neutralizing and total antibodies were fluctuating in these Ebola survivors. So they would go through periods where their antibody levels in blood would be sort of consistent, and then they'd go up and surge down. They would surge up and go down and surge up and go down. And what the only conclusion that the team could really come to was that the antibodies were probably surging during periods in which virus that was probably viable, real virus, in these anatomical sanctuaries might have been reactivating during short periods and causing an antibody response. So that sort of began to ask people to say, like, maybe that's virus. And then what happened is a couple months ago, a new Ebola outbreak started in Africa, and several independent teams tracked the genome of the Ebola virus that started that epidemic, and it was virtually identical to a strain that had started an outbreak 5 years before. So most of the teams concluded off that analysis that it was chronic Ebola that must have been transmitted by a person infected years before, who started the outbreak. And now it's interesting, the post-Ebola community is actually basically looking at this as a chronic infectious disease. Now, that's interesting is the post-Ebola patients, if they cough on you, they don't give you the Ebola. It's really hiding out, right? So, you know, that comes into all these with long COVID. Like, let's say someone has SARS-CoV-2 in a tissue. Are they contagious? Probably not. But, you know, so again, we need more research on this. So then, Charlotte, going to your question of has that been researched in long COVID? No, except there was not yet, except there was one team, and I forget where, that looked, not in the US, but they looked at penis tissue from men who were undergoing erectile dysfunction surgery. So they had gotten erectile dysfunction that started after COVID, and it got bad enough that they got surgery to address the issue. And when they analyzed, I forget how many of the penis tissue samples, they found the virus in at least several of them. So it had reached there. Yeah. So that suggests we should probably do, I don't know, some studies of semen. I don't know. Maybe.
Thank you.
Wow.
But does it mean potentially? Can you hear me?
Yes.
Yeah. Does it mean potentially that it will become a sexually transmitted disease?
I really hope not. Honestly, I don't want to scare people with this. I do think that it's possible that the post-Ebola syndrome, the persistence in the sperm, I almost want to hope that was like an Ebola thing. I don't know. They have found, for example, somewhat similarity in Zika. Even then, those patients didn't necessarily always sexually transmit. It seems like when virus hangs out, it really isn't active most of the time. And again, we really need to, RNA viruses are not studied well for persistence. We study the DNA viruses like herpes viruses. We acknowledge their persistence. We study the mechanisms more. We study what they do. We study their forms of latency. But RNA viruses, such a lack of research into RNA virus persistence. So we know there are certain mechanisms. For example, some viruses, RNA viruses like enteroviruses, which can also cause common cold or respiratory symptoms, they can form into what's called a non-cytolytic form, where they actually like lose a little side of their genetic material, and that helps them embed in a cell and kind of stick around there in like an equivalent of latent form. And so when they're in that form, people don't think they're contagious. They don't think that it's just a way of hanging out. So, there may be, but then they can activate, right, under certain conditions where they can act more like virus, right? So it's a little crazy. They can kind of fluctuate between being able to be transmitted, being able to do things, being able to move, and not. So, I don't want to scare anyone with this, with the Ebola cases and all that, it's a thing, but I think that with long COVID, we got to look at the samples. We have to do this. But I have to say that if it was blatantly sexually transmitted, we might have heard more stories by now. I don't know.
Let's hope.
Yeah.
Yeah.
But of course, it's all very complicated and hard because just as like you were saying, that most of the people, like 80, 90, whatever percent, they have, for example, mono Epstein-Barr, right?
Mhm.
So, I mean, even if you find it in some tissues, 90% have it. So you cannot just conclude that, okay, this is causing this and this. Even herpes, herpes 1, how many people have this on the lips, right?
You're
So
totally
it's very
right.
hard.
It's really hard. You're exactly right. That the presence or absence of a virus isn't necessarily, it's important to know, but it's not always the most important question, at least to me. The most important question is, what is it doing? In other words, what is its activity in that given patient? Because exactly, you can have a person who has Epstein-Barr virus and it's controlled by the immune system and it's just chilling. Or you can have Epstein-Barr virus quite literally drive tumor progression and cause cancer, and that can lead to death. And what happens is in the cancer patient, Epstein-Barr virus begins expressing different proteins. It expresses multiple proteins that begin to change the metabolism of the cells that infect and directly hijack human gene expression and directly change, stop cell, replication inhibit. It starts to totally act differently, and the proteins that it produces are different. So it's not just a question of that's the thing is, we first need to know if we find the SARS-CoV-2 virus in patients long term, but then we really do have to say also, what is it doing? Is it still expressing protein? What proteins is it expressing? What is going on there? What is the gene expression of the cells that it has infected? And what's cool is some of the computer-based tools that we use allow us to do that. We can actually take a single infected cell and we can figure out all the genes that a virus is turning on and off. And that may be very different in a sick person than in a person who is not as sick. So good point.
And since 90% of the world's population have Epstein-Barr, or more or less, then it means that there are many other conditions necessary for it, for example, to coincide, to drive it to cause cancer, right?
Yeah, one of the things that people are looking at in, for example, conditions that Epstein-Barr virus can contribute to is, it's a component of a mix that can add into cases, right? It's, polymicrobial disease is a growing concept in which you might have Epstein-Barr virus and you're okay. And this really, really pertains to long COVID. You might have Epstein-Barr virus and it's controlled, but then you get a different infection. That infection, downregulates part of the immune system and allows the Epstein-Barr virus to express more proteins and become more problematic. Or it doesn't have to be an infection. It could be a chemical exposure. It could be exposure to mold, which can knock down the immune system. And now that allows the virus to be more active, more problematic, right? Or it could be a difficult pregnancy or an accident, which, causes a lot of, problems. So people can go through sort of what we call a multiple hit model, where different events happen to them and different things happen. And that sort of leads to a collective process in which different pathogens and different issues can sometimes compound themselves. And depending on the genetics of the patients, the microbiome that they have, other factors they have, the Epstein-Barr virus will head in different directions. It might end up causing cancer. It might end up doing something different. It might end up doing nothing, right? So in other words, we have to study these viruses not in isolation, and we have to, like Steve mentioned before, start to consider the integrated impact of everything happening in patients, from the human genome side to the microbiome, virome, to these pathogens, to other aspects of disease, to really understand the end point of disease that any one patient develops.
Yeah, I understand. But Epstein-Barr is very common, right?
Very common, yeah. I would say, 90% of people have Epstein-Barr virus. You can also, it depends on the way you look at these viruses. a really interesting line of research now in Alzheimer's disease is viral contributions to Alzheimer's. And there's a team at Harvard that's doing research that indicates that the plaque in Alzheimer's brain, the amyloid plaque, may actually have a function. It may actually function as part of the immune response towards pathogens in brain tissue, right? So it's really fascinating research, in other words, that Alzheimer's itself may have an infectious component. And so one of the things that can happen is patients, a lot of people harbor Epstein-Barr virus, for example, and they may be okay for most of their lives. But then as people age, you go, why do you get achy? And why does your functioning, I'm actually part of a couple anti-aging groups because there's not that many differences, for example, in what we call aging sometimes in chronic disease. So you may harbor Epstein-Barr virus may play into processes that we just deem aging. If you're 75 years old and you say, I feel achy and worn down and fluey, everyone goes, you're old. That's normal, right? If you say that when you're 30 years old, they go, oh, you must have a disease, right? So there's all kinds of ways in which these viruses, we all have them. A lot of us have them, but they may contribute to, God knows, aging processes or things in ways that could be potentially detrimental to the majority of people over the long, long, long term. I don't know.
Yeah, makes sense, of course. But on the other hand, if 90% have it, it's not that all of this 90% will get older or feel older sooner, right? Because they are so common. So it's such a complex process.
Yeah. No, I mean, it really depends on a lot of factors. I think some people can just, handle the virus, right? But would, I don't know. The fact is, like, if you take any condition now, multiple sclerosis, Parkinson's, the Crohn's disease, the GI conditions, you'll start to see more teams study them as conditions that involve pathogens, including the herpes viruses. So harboring these viruses may actually feed into more of the chronic disease that we see across the board that the majority of people develop. I think in the USA, 65% or more of Americans by the age, or I forget what the proportion is, Americans age 65 and older, I think, have like at least 2 or 3 comorbid conditions. I mean, most people have something, right? By the time they've reached age 65. And so you have to start to wonder, like, right, a lot of people have Epstein-Barr virus, for example, but a lot of people get chronic disease. So, it's a ongoing research question.
Thanks.
Yeah, no problem.
Thank you, everybody. This was an excellent panel. Dana had to log off. Does anybody else have anything? No, we're good. Well, thanks everybody for your time and attention. And feel free to hang out, network, chat, and this recording will be available through the World Health Network, and we'll be in touch with everybody. Thanks.